These are the recorded lectures for Chapter 3 on cell structure and function. This is another one of the chapters that is a review chapter where you're going to see a lot of this information throughout Bio 160, throughout the course. And also, you're going to see it on some of this information on the TEAS test. So we try to always cover the chapter on chemistry and the chapter on cells, especially for that reason. It's an important chapter, but it's not like one of the body systems. So I try to explain it to students that this is why we're doing this, Now, you've got your learning outcomes listed. I'm going to just go through them. You can read those on your own if you want to, but we're going to start with the first thing you need to know about cells is the cell theory. This is a theory that was developed by many biologists, some that studied plants, some that studied animals. We call the ones that study plants zoologists, and the ones that study, I said it wrong. I'm so sorry. I should just redo this, but I'm not. Botanist. Plants are botanists and animals are zoologists. But anyway, many different scientists looked at cells under their microscope and developed the cell theory. And basically what it says is that all living things are composed of cells. So part one of the cell theory is that cells are the building blocks of all plants and animals. Part two is cells are the smallest functioning units of life. Part 3, cells are produced through division of pre-existing cells. So this is what this means is that there's not going to be any living cells that are generated by magic or by spontaneously generating, anytime you have a new cell, it's because an old cell or a parent cell divided and produced the new cell. So pre-existing cells divide and make new cells. And each cell maintains homeostasis within the cell. Okay, so the human body contains trillions of cells. And to elaborate on that, I'll just let you know, let's say that on average, a human has 100 trillion cells. So only 10 trillion of those cells would be part of the human body, would actually be cells of the human body. 90 trillion would be microorganisms that live on or in the human body. So sometimes that's fascinating to people, to find that out. Now, the study of cells has a name. It's called cytology. Cyto means cells, L-O-G-Y means study of. So cytology is the study of the structure and function of cells, and that means Chapter 3 really can be titled cytology. What we use to study cells are microscopes. We use light microscopes in the lab, and they will magnify up to 1,000 times bigger than the cell that really is. And then we have electron microscopes, which are used. They magnify, up to tens of thousands of times, and they are used to see detail. These are some examples of different cells of the human body, so you can see the differences, but also the similarities. If you look at all of these cells besides the red blood cells, you can see a nucleus in each one. This is a smooth muscle cell, and even though it's kind of tapering on the ends and it's thin, there's a nucleus in there. These are cells that line the intestinal tract, and they've got a nucleus here. They're tall cells that have these folds called microvilli on the surface, and they have a nucleus that's kind of near the bottom of the cell. The bone cells have these extensions, and there's the nucleus. A neuron, here's your main cell body with the nucleus, and then it's got extensions to receive signals and then an extension to send signals. A fat cell is mostly filled with fat. That's this yellow portion, and under the microscope it would look white. But it's also got a nucleus kind of pushed to the edge of the cell because of all the fat. And this is an egg or an ovum. Here's your nucleus. This is a sperm. So these are just different types of cells in the human body. But all cells, what all cells have in common is a plasma membrane, and the plasma membrane separates the inside contents, which is the cytoplasm, from the extracellular fluid, which is what surrounds all of our cells, except for skin that is exposed to the environment. You know, like if the skin of your, I'm just looking at the skin of my hand and my arm right now. That, of course, is not surrounded by extracellular fluid on the outside. It's surrounded by air. But most of our body cells are surrounded by extracellular fluid, which is the fluid on the outside of the cells. We also call it tissue fluid or interstitial fluid. This is a model of a cell. And this one, well, let's just blow it up so we can see everything. All right, so first we have, in this model, it's showing you the plasma membrane. Okay, so the plasma membrane surrounds all of our cells and separates the cytoplasm from the extracellular fluid. The cytosol is the part of the cytoplasm that is the fluid between the nucleus and the plasma membrane. So this is the cytosol, and then we have, it's showing you the centrioles, which are structures that function in cell division. The cytoskeleton, you see all these fibers everywhere, all these little fibers. Some are purple and some are look kind of brown, you know. But the cytoskeleton are protein fibers that play different roles in the cell, help it maintain its shape. They help keep the organelles in place. Some of them function in movement. Now, let's see the next. Oh, I have to make it smaller. All right, so here you also see some structures like, I guess I need to blow it up too. This organelle is called a Golgi apparatus or a Golgi complex. And it's just, it's going to package up the products of the cell. If the products are usually either some kind of protein or some kind of lipid or fat or carbohydrate, but it will package up the products of the cell into one of these little vesicles. And they pinch off the ends of that Golgi complex. So here's the Golgi, it's for packaging. We also have, here's our nucleus. Inside the nucleus is the nucleolus. The job of the nucleolus is to produce ribosomes. And you can see the ribosomes, they're either floating around free or they're attached to the endoplasmic reticulum. This endoplasmic reticulum here is called rough ER because it does have ribosomes attached to it. Now, if you come a little farther out, you'll see the smooth ER. See, this does not have ribosomes attached to it. It's a little bit further away from the nucleus. The rough ER is close to it, and then you have the smooth ER. So the rough ER produces proteins, and the smooth produces lipids. And then you have these blue structures. These are mitochondria, and they carry out cellular respiration. They produce ATP, energy for the cell. So we said ribosomes produce proteins. Peroxisomes function in breaking down fatty acids, mainly. They break down fatty acids. Lysosomes function in cellular digestion. And then here we have just a little bit more about the Golgi apparatus, storage, alteration, and packaging of secretory products. The mitochondria produces 95% of the cellular ATP. The endoplasmic reticulum, it shows you the rough and the smooth. Rough synthesizes proteins. Smooth ER synthesizes lipids and carbohydrates. And then there's a lot of information about the nucleus. So this is just the structure of the cell. The study of cells is called, and that is cytology. If I can write it, I will. I will try. Oops. Cytology, study of cells. Okay, I did it without a crazy marking everywhere. Functions of the plasma membrane. There are 4 functions listed here, but mainly it regulates exchange with the environment, because anything that has to come into the cell has to cross the plasma membrane, and anything that has to leave the cell has to cross the plasma membrane. So another thing that we learn about in this chapter of cytology is transport across the plasma membrane. And this is that section. So the structure of the plasma membrane, you can see here that it's composed of this double layer of, these are called phospholipids. So here is one phospholipid. It's got the head is a red color, and then it's got 2 tails that are kind of purplish colored, but there are 2 layers of those phospholipids, and we call that the phospholipid bilayer, the phospholipid bilayer. And then in addition to the phospholipid bilayer, you also have these large blue molecules, or proteins, and we have other lipids like cholesterol, and then we have carbohydrate chains that are attached to our phospholipids and sometimes attached to our proteins. But this is mainly how the structure of the plasma membrane is: a phospholipid bilayer with proteins embedded within the phospholipids. And this is a larger image that shows you a lot more detail. You can see the phospholipid bilayer. Okay, so you can see the phospholipid bilayer, double layer of phospholipids, forms the majority of the plasma membrane, but then embedded within the phospholipid bilayer are these large blue protein molecules. This one is a channel protein that allows molecules to come in and to leave the cell through this channel. This one is a carrier protein. Well, actually, it says it's a protein with a gated channel, so there's actually a gate that will open in response to a chemical or change in difference in charge across the membrane, things like that. Something will open that gate, and it will allow molecules to move in or leave the cell. Then we have cholesterol embedded within the phospholipids. And then we also have these chains, these carbohydrate chains. And if the carbohydrate chain is attached to a phospholipid, we call it a glycolipid. And if it's attached to a protein, we call it a glycoprotein. So here are the notes on the plasma membrane. Phospholipids are the major component of the plasma membrane. And remember, there's a double layer of those phospholipids. So it's a phospholipid bilayer. Cholesterol gives stiffness to the plasma membrane. Materials that can cross through the phospholipid bilayer and don't need to pass through one of those channel or carrier proteins, there will be lipid-soluble materials. So as long as they're soluble in a lipid, and some examples of that would be oxygen gas and carbon dioxide gas. But if they're lipid soluble, they can cross through the phospholipid bilayer easily. If they have a charge, or if they are polar covalent molecules, that would be ions. That means they have a positive or a negative charge. And if they're water soluble, then they have to cross the phospholipid bilayer. They can't cross directly through the phospholipids, but they have to cross through one of those proteins. Now, the phospholipid bilayer has a hydrophilic head, and it also has hydrophobic tails. And the way the phospholipids are arranged, you can see that the hydrophobic tails face each other, and the hydrophilic heads face away. And so the hydrophilic heads are water soluble, because hydrophilic means water loving. So they're going to face out to the extracellular fluid, and then they're going to face inside the cell to the cytoplasm, where they're in contact with a watery environment. And then the hydrophobic or water-fearing tails are going to face each other so they don't contact water.